Sorry about the delay there. I'm still trying to get the DVD for Lambda to work, which I'll also post in the library. We'll see how it goes. Of course, it worked perfectly fine on this computer in my office right before I came over, as usual. Exams have been graded. All the scores are posted on D2L. I do not have your Scantrons with me. I will also talk about some of the questions on Friday in lecture, and I'll try and bring the Scantrons at that point as well. So if you have any questions about specific questions on the exam, please hold those until Friday, and then we'll talk about them. There's a distinct possibility that something got scanned incorrectly, you didn't erase your answers properly on any of the particular Scantron questions. If that's true, let me know. I've actually got copies of all of them. I scanned all of them. So that's there, and we can certainly take a look at that. It happens, and so we can just take care of that. So please let me know. Just send me an email about that. Any more questions about the exam? Also, high score is 41. That will be scaled from that, as usual with my exam. So 90% plus is an A, and so on and so forth. It's all in the syllabus, etc. And I'll post those as well, that breakdown. That's the minimum score, basically, that you would expect to have an A at. That number will probably go down a little bit towards the end of the term. Okay, some people took the exam, and I don't have any clicker scores for you. If you've been using a clicker and you think you should have scores, just send me your clicker number and your Odin username, and I will make sure that that gets updated and posted. I know that one or two of you actually just emailed me today, and I haven't had a chance to upload those yet. Okay, other questions, comments, worries? Strangely enough, a lot fewer people here days we don't have exams. That's your choice, not mine. So today we're not going to start with kicker questions because we kind of did those already, but there will be some later, don't worry. So today we're going to talk about Lambda phage, and I had some complaints earlier that we're only going to talk about Lambda phage for one day, and Patrick's not even here, even though he's the one that really wants to hear about it, that I'm sure we'll get here. So phage Lambda is, as some people have called it, I think actually the name of the chapter is a pinata of paradigms. So really just an amazing amount of work has been done on bacteriophage Lambda. Everything you need to know about bacteriophage Lambda is on this poster, which is now falling apart. We need to scan and actually get a nicer copy of. So phage Lambda, lots of people also in San Francisco in the 60s were working on this. You can kind of tell by the color scheme. This will be up here. You can kind of take a look at it a little bit later on. But it's a really nice overview of what's going on in this process and drawn out much nicer than I ever could for any of these things. So we're going to very briefly cover sort of the highlights of what's going on with Lambda bacteriophage. These are double-stranded DNA bacteriophage, and again, paradigm, blah, blah, blah. But the main thing in terms of the paradigm really has to do with transcriptional regulation and then transcriptional regulatory circuits in terms of how Lambda is functioning. And really the big thing there has to do with the decision between whether to blast open your host cell or just hang out inside it and just be there. And let the cell do all the work and let yourself replicate as it moves along. And this is true for Lambda. Almost all bacteria, almost all archaea, all have these virus genomes sitting inside their own genome. What about our genome? Loaded with virus. So it does seem to be, this is a very common practice and sort of originally understood in looking at Lambda, but probably true for pretty much all organisms. We've all got virus genomes inserted in us and pretty much everything else as well. So it's just that understanding process that we know about here, but it's very, very general. And then how that happens is really, again, through integration of the virus genome into the host genome. And again, best understood for Lambda, but true for many, many, many other ones. Couple of important aspects here, sort of key concepts, things to think about. Were they useful looking at key concepts for the exam? Yes, no, maybe, kind of, sort of. So a couple of things as far as Lambda that are very important and things that we haven't really talked about so far having to do with anti-termination, and this is anti-transcriptional termination. Turns out that really, particularly the steps on the road to lysis have a lot to do with transcriptional anti-termination. Feedback loops, this is a classic example, in fact probably the first example, thinking about how expression of certain proteins can lead to feedback and basically a switch. Once you've had the classic genetic switch, the other book that I could have brought was the genetic switch. It's a lot skinnier than this, but also thinking about how you have changes that happen at one point in development, at one point in virus infection, that then get maintained. And again, this genetic switch is really, do we go through lysis and blast open our host cell, or do we go under lysogeny? And then, of course, integration is the last point here. Yeah. Oh, where? Lysogeny? Oh, lyosgeny. Yeah. Okay, that'll be one of the questions on the exam. How do you spell lysogeny? No. No. Thank you. I will try and repair that. This one's right. Lysogenic growth. So yeah, we'll talk a little bit where Lambda came from. Lambda was basically discovered by mistake, and true with many things in biology. Lots of mistakes which turn into really good things. There's a new book that just came out. I don't know if any of you have heard about this. They talked about it in This Week in Virology this week, called Failure, and why it's critical in science. So a lot of that failure, mistakes, etc., accidents. But a little bit about the structure. The structure is relatively boring for bacteriophage Lambda. It's got a nice icosahedral head, cool tail structure. Binding and entry, again, relatively straightforward, although a lot of what we don't know about Lambda actually has to do with this entry phase and how it actually gets its genome inside the cell. So unlike T4, T7, and to some extent 5174, we're not quite sure how it gets its genome inside the cell. But the big deal, of course, is transcription. That's what we're going to spend most of our time talking about today. Really the two different pathways, lytic growth and lysogenic growth, both of which are really regulated at the level of transcription, and then how you make that decision between the two. Talk a little bit about replication and maybe something about release, assuming that I can get the DVD to work at the end today here. Worst comes to worst, I can put it in the other player here. So where did it come from? Again, this is one of those oopsie moments. People were working with E. coli that they thought had no viruses associated with it, and then lo and behold, they were mixing together two different strains of E. coli, and all of a sudden all these plaques started to appear on different strains of E. coli. And then they went back and checked, and it turns out that what was happening is that there was a prophage, and the whole idea of a prophage, provirus, it's a viral genome integrated into the host genome. So this standard strain of E. coli K12, had a lambda genome in it. And actually there are about 7 lambdoid genomes in the standard E. coli. This is the only one which is in fact completely active. So people have been working on this strain for 30 years and didn't even know it was there, which is really pretty amazing. And again, is typical of how science progresses. Yeah?
Do you mean, when you say 7 lambdoid, do you mean 7 different types of lambda genomes?
7 different kinds of lambda-like genomes present in the host, yes. And so, yeah, these plaques, again, formed on a mutant of K12, which actually turned out to be missing lambda. And we'll talk a little bit about why that has to be the case later on. And the reason they didn't find it is that once you have one of these prophages, so the integrated form, you're resistant to infection by new bacteriophage, or at least bacteriophage of the lambda sort. We'll talk about how that works in just a second. But it was, once they found it pretty normal, about a 200 burst size, so 200 virus particles produced per infected cell. But the big difference, of course, here is that this is a now integrated form of the virus. And this is the first one that people had really found to be that case. And so basically what they had was E. coli that was like this. The virus genome, which is this little sort of brownish thing in the black cellular genome. Now, what we now know is that this whole process starts out, got our nice virion, the genome comes inside the cell, and then you make this decision, the genetic switch. Either undergo lysis, where you make a whole bunch more of the genome, it makes all the viral proteins, you make virions, it lyses the cell, etc. So this is kind of what we've talked about already, this whole process right here. Lysogeny, on the other hand, is when the genome integrates itself and the cells just hang out and replicate these genomes. And again, this is true for almost all genomes that we know about today. They have these proviruses or prophage, in the case of bacteriophage, hanging out in the genomes. And just one real quick part of terminology here, a lysogen. There's a lot of confusion about whether is lambda a lysogen? No, lambda is not a lysogen. The E. coli K12 with lambda in its genome is a lysogen because it can generate a lytic event. And so lysogen, generation of lysis. So that's the E. coli lysogen here. Any virus that can do this, which is then hang out in its host instead of lysing its host, is called a temperate virus or a temperate bacteriophage. So the virus is temperate. The infected cell with the provirus in it is a lysogen. Does that make sense? And then the last thing that we'll talk about here is how you do that. So once you're already in the genome, at some point, the virus needs to make more of its genome and capsids, etc. So this is the induction phase. And we'll talk all about these different processes as we move through here. So there's your lysogeny and your lysogen. So let's look at what happens, what comes into the genome, into the host, I should say. The lambda genome is really kind of interesting because it is packaged inside the virion as a linear genome, but as soon as it comes inside the cell, it circularizes. And we'll see, again, hopefully if we can get the video to work at the end, why the linear genome packaging makes a lot more sense. But in terms of actually undergoing replication, circularization makes a lot of sense in terms of transcription. Circular genomes are way better than linear genomes. Linear genomes are just obnoxious and annoying. So circularization, hey, that's what I've got. No, I haven't managed to circularize any of my chromosomes. But yeah, it makes a lot of sense in terms of what's going on there. But interestingly enough, these were things that a number of people whose names should sound vaguely familiar have been discovering. Hershey, as in Hershey Chase, was also looking at how Lambda was functioning. Alan Campbell, there's a really nice video of Alan presenting some of the work that they've done on the integrase gene in the phage birthday party that I was involved in last year in 2015. So there's a whole, he's got about half an hour where he talks about how he thinks Lambda is regulated. And best part about it, it's like chalk talk. Everyone else does PowerPoint, he's got chalk. And so he showed that site-specific recombination was going on. And in fact, Alan Campbell reviewed our paper that we did with our virus genome. We knocked out the integrase gene and said it was a load of crap. Still got published, but it was pretty impressive to have this guy looking at this. So genome, as I mentioned before, double-stranded DNA. It's a tiny bit bigger than bacteriophage T7, but similar kind of size. And hopefully you remember from bacteriophage T7. No, we don't remember bacteriophage T7. That was on the last midterm. It's got a lot of its own machinery. And so very similar to the case of what's going on with bacteriophage T7. But it turns out that Lambda is actually using more of the cellular machinery than T7 does. Almost 50,000 base pairs in length, and it was one of the second complete genome sequences to be determined, also by a guy by the name of Sanger, which sounds vaguely familiar. So it's like name dropping in terms of molecular biology when you start talking about Lambda. One of the things they found with this genome, first thing was that at each end, here where you've got these so-called COS sites, COS just stands for cohesive ends. These are 12 base sequences that are present at each end of the genome. which are complementary to each other. And so that's how you get circularization once you come and get inside the cell. Al Hershey didn't know this. He just saw that circularization happened. And it was only after the genome sequence that people saw these 12 base ends, which were overlapping with each other. And also, of course, as soon as you have the genome, you can start to look at where all of these genes are. And it turns out they're all put together in, as I think they mentioned in the textbook for T7, as an engineer would do. You know, put the various different gene blocks together on what you need to have. So all the head genes are together, all the tail genes are together, tail fiber genes are together, recombination genes are together, etc. And so we'll go through and look at some of those in a little bit more detail as we go through this. The lambda structure is a nice T=7 head, and for a nice exercise, try and find the T=7 on here. It took me about 20 minutes looking at this to try and find all of these, so I wouldn't be asking you to do it. Also has this flexible tail structure, and this is really quite different than a number of the other bacteriophage. And one of the ways that people classify bacteriophages is to have a rigid tail or a flexible tail. And it turns out for Lambda is one of the most flexible ones. There are these tail fibers present here at the end. This is in fact not in your textbook. These tail fibers, interestingly enough, even though people have been studying bacteriophage Lambda for over 50 years, they only found tail fibers about 10 years ago. Turns out they were working with a mutant that didn't have the tail fiber genes. So the Lambda bacteriophage binds to a particular receptor. It's a maltose transporter, but because everybody was working on Lambda before they were working on how bacteria E. coli grow on maltose, they actually identified it as the Lambda binding site as opposed to a maltose transporter. So the maltose transporter is called Lambda. The reason the maltose transporter is called Lambda is because it's important for Lambda binding. And as usual with all geneticists, what do? You break something, and then you name that gene on what the phenotype is of the mutant. So Lambda binding, have a mutation in that Lambda, you don't have Lambda binding anymore. Also means that when you try and grow Lambda on E. coli, you also have to include maltose, because include maltose to induce the expression of the maltose transporter gene. So a lot of people have trouble growing Lambda. They'll ask me, I can't grow my Lambda. I say, well, yeah, you're growing maltose? Oh, yeah, I have maltose in the media. Yeah.
That's the tail binding to that transporter, not the fibers.
Yeah, so it seems to mostly be this tail, and particularly the tail cone, that we'll take a look at in just a second. So they're very direct interactions here with this tail protein. The fibers are probably important for getting the genome inside the cell, although they're also kind of dispensable because a lot of these mutants work just fine. People use them in the past. And again, circularization of the genome, which we've talked about already. So let's first start talking about the easy stuff. And what I've tried to do here is include sort of an overview of that cartoon from right at the beginning. And we'll talk about each of these different areas. So we'll talk about lytic growth first, then we'll talk about lysogeny and a little bit about this induction process. Lytic growth is the easier one to talk about. And the main thing as far as transcriptional regulation of bacteriophage Lambda has really to do with this little part of the genome. Right here, this is the regulatory part of the genome, and it's just blown up down here. And I've tried to include all of these extra images here for cross-reference. So what happens in lytic growth is you have expression of, surprise, surprise, immediate early genes. And these immediate early genes, just like the case that we've talked about before, are going to be regulating host functions in order to get something to happen in terms of the viral genome. Again, incredibly creatively named are the leftward promoter and the rightward promoter. And again, that's based on the way the genome is set up here. So going to the left, our genes are being expressed in this direction. Going to the right, our genes are being expressed in this direction. And one thing to remember is this is now a circularized genome. So once it's inside the cell, it's circular. And what happens is you have expression from PL, expression from PR. You make the CRO protein. CRO stands for control of repressor and other proteins. And we'll get back to the control of repressor a little bit later on. N is just a gene that they found really early on and only later found out what the function is of this particular gene. So the N protein is absolutely critical for getting lytic growth because N is an anti-transcriptional terminator protein. So in the absence of N, that's what this minus N here is supposed to stand for, you have termination of transcription at this terminator, L, and this terminator, R. If you make enough N protein, then you have anti-termination, and so you don't terminate here, and you keep going. And so that's what's here shown with these longer transcripts here. You also have these two sites, and these are RNA sequences, which are NUT sequences. What's NUT stand for? It's N utilization. So if you have the N protein and one of these NUT sites, then N can serve as an anti-terminator. And N just binds to the RNA and the RNA polymerase in order to stop the termination from taking place. So here we've got continuation past this leftward transcript, and we'll see that actually goes off here. But much more important in terms of lytic growth is concerned is what happens at this point. We've got anti-termination that happens here. You make the O and P proteins, we'll see later, are really important for replication. But in terms of the rest of transcription, the Q protein, which is just like the N protein. It's another anti-terminator. And so this anti-terminator protein is most important for transcription of all of the rightward genes here. And the important thing on the rightward genes is not so much those here, but all these that are over here on this side. Because remember, they're circular. And so all of those are going to be your structural proteins, head proteins, tail fiber, and lysis proteins as well. And so that's all now what's happening in the lytic process. So again, that's the easy thing. It's all about anti-termination. Okay, happy with this? You can see he's reaching for his clicker. No, we're not happy. More questions?
Okay, so yeah, I need to clear really quick. So it comes in and it initially starts doing these early gene transcription translation, right?
Mm-hmm. Oh, the immediate early ones, yeah.
The immediate early.
Mm-hmm.
And this N protein is one of the early things to be translated, transcribed, translated.
Correct.
And it sort of is binding to some and creating like a positive effect while simultaneously doing a negative. Is that what that's showing?
Okay, well, so it's a negative of a negative, maybe is one way to think about it. So it's an anti-terminator. So termination.
Demonstrating an absence and presence of this N repressor.
Okay.
Happy? Yes? No?
So
Kinda,
if you
sort
don't
of.
have N, the show ends early.
Okay.
Have N,
Okay.
it goes.
Yeah.
All right.
Okay. Did people hear that? So don't have N, party stops. Have N, party goes.
Okay.
Yeah.
Well, I was just wondering, budding viruses less organized. Do they like?
Oh, so yeah, the whole idea of budding and lysogeny.
Yeah.
Yeah. So as far as the literal definition of that, anything that's budding and actually not lysing the cell is not going to be a lysogen because you're not lysing things. So by definition, no, it wouldn't actually be a lysogen.
Is there a incorporation into the cell genome?
So, but you can still have a provirus in the genome, and that, as we'll see, is true for almost all the retroviruses when we get to that point much later on. Okay, so can I finally do my clicker question, please? Regulation of expression of late genes in Lambda is mostly due to regulation of transcriptional initiation, transcriptional elongation, transcriptional termination, translational initiation, translational elongation. And I don't think there are any typos in there. 27. 27. Mean. Yeah.
So, okay, and K is a
Well, of 41. Yeah, you can do that. Yeah, do that calculation. Yeah. 10. Four.
yeah. I mean,
Yes, it's about anti-termination. A couple quick things about this particular question. First one is, what the heck are late genes? Those are going to be all your structural genes. And so those structural genes, the head genes, the tail genes, etc. And they're all going to be dependent on the expression of Q. Exactly, which is a anti-terminator. Exactly. Okay, so let's look at the other side of the equation, I guess, is one way of thinking about it. So what's going on now with lysogenic growth? And how does lysogenic growth happen? And we'll talk a little bit more about this later and how the virus knows to totally over-anthropomorphize these things if the host is unhappy. So if the host is unhappy, and basically the host being unhappy means that there probably aren't a whole bunch of other uninfected E. coli to go and infect. So it's not to the virus's advantage to make a whole bunch of new virions. It's probably to their advantage to hang out. And if you think about most life, we don't have ridiculous amounts of resources. In fact, we're all resource limited, I think, in lots of different ways. So the hosts are generally not going to be very happy. You just have to detect that, however, in one cell and assume, again, to anthropomorphize, that all the other cells are under similar kinds of conditions. So you're not growing really fast. So if that's the case, then what you do is you make the C1 protein. And C1, I think it makes it a lot easier to understand. The C1 is just a horrible name. But again, it's from the geneticists. And so the geneticists found these particular mutants. And this mutant in Lambda was the clear mutant. So clear, what does that mean? That means you have a clear plaque. And that clear plaque means that you get lysis. And you get lysis all the time. So your C1 is a mutant that causes lysis all the time. And so what it's doing normally is helping you get lysogeny taking place. So what happens in the presence of C1 is that C1 binds to, the operator, and operators hopefully mean something to you based on lac operator, operons, etc. These are just binding sites for regulatory proteins that just so happen to overlap with our friend PR and, not shown, PL. So PL and PR, those are the ones that are making CRO and N. So in the presence of Lambda C1, the Lambda repressor, it represses transcription from PR and also from PL over here on the other side. The way that works is the Lambda repressor binds really well to operator right. Again, 1, there are 3 of them. Better than 2 and not so well to 3. So this process is also a highly cooperative process, meaning that if you have binding by 1, in this case dimer, almost always true of regulatory proteins, we can talk more about that later, dimeric regulatory proteins, binding of 1 dimer stimulates the binding of the second dimer, and it's these guys together which lead to repression of transcription from PR. Interestingly enough, Lambda repressor, and that's why Stedman has it up here in quotes, is of course not just a repressor, it's also an activator. And if you have binding of the Lambda repressor to OR2, that stimulates transcription from this promoter here, which is the promoter for, what does RM stand for? Repressor maintenance. So it's getting more, sorry, I cut you off. So it's all about making more of the repressor. So this is the first feedback loop that I'm sure all of you remember from molecular from last term, where the presence of the lambda quote-unquote repressor leads to expression of the lambda repressor. So you get more of the lambda repressor, which leads to more expression of lambda repressor, etc. That only goes to a certain point, and that is when you finally get binding to OR3, and the same thing is true of OL3 right here, these orange versions of the lambda repressor. That then will also block transcription from here, but that means you've got a lot of the lambda C1 protein around, and that completely then blocks things. So this is in lysogenic growth. Of course, the question is, how do we get the lambda C1 protein in the first place? We'll get there. But I wanted to talk first about prophage induction, which is the process here where you go from being in this happy state in the positive feedback loop that you have from lambda C1. How do you escape from that positive feedback loop? It kind of seems like the black hole that you're falling into here. So the way that works is you've got to get rid of lambda C1 somehow. And that process is the induction process, goes through something called the SOS response, or the save our souls response, ... Mean anything to anybody? I see at least one nod here. Save our souls. So basically, if things are going really, really, really bad for the cell, and that's usually detected by DNA damage, and the classic way that this works is ultraviolet irradiation forming pyrimidine dimers, and the formation of pyrimidine dimers turns on a big DNA damage repair response, and the virus, which has been hanging out, tanning itself appropriately in the genome, says, this might mutate my genes too. I better get the heck out of Dodge. So the way that happens is it takes advantage of this SOS response, and part of the way the SOS response works is there's a cellular transcriptional repressor called LEX-A, which gets cleaved and leads to the whole SOS response, and C1 has evolved to be really similar to LEX-A. And so this cleavage that happens by LEX-A under the SOS response happens in a very similar way with C1. Once you have this cleavage that happens, so C1 gets broken in half, then it no longer represses PL and PR, and then you make N, which makes Q, which leads to expression of all the lytic genes. So that make lots of sense? Yes? No? Good? No, it's not a clear question yet? So, of course, the big question is, how do you get C1 in the first place? And so that's the whole idea of the genetic switch, and again, the book by Mark Tashney. How many went to Mark Tashney's talk over at Reed? Really obnoxious. The guy's completely full of himself. But smart guy, too. But he came up with this sort of idea here with the genetic switch. I should probably erase that.
He says the same thing about you.
No, in fact, no, he's the first one to admit it. There's also this great thing circulating. I have to try and find it again somewhere, which is called The Gospel According to St. Mark. And St. Mark Tashney about the way that all gene regulation is supposed to happen this way. So I'll see if I can find that for you. So basically, has to do with C1, we already talked about. And then this protein CRO. And again, CRO stands for control of repressor, i.e. C1, and other genes. But most important for us is just really about the control of repressor. So it's all about how much CRO you have versus how much C1 you have. Because CRO basically binds in an opposite way to C1. So CRO binds really well to OR3, which is what C1 binds the worst to, and not so well to OR1. On the other hand, of course, C1 binds really well to OR1 and not so well to OR3. Same thing is true for operators at the left operon. So here, it's all about high levels of C1, then PRM, which is your repressor maintenance, but most importantly PR, which is what's going to lead to lysis or not. Complete absence of C1, PR is on, you get lysis. In the presence of C1, PR is off. Crow, if you've got small amounts of crow, then you're going to have some expression of this. As soon as you have much crow, that's going to shut down PRM and allow this regulation of PR. So, okay, the next question is, well, how do you regulate between amounts of C1 and crow? Well, that comes down to how you make C1 in the first place. I just talked about PRM, but PRM is the promoter for repressor maintenance, not for getting the repressor in the first place. And so that is the promoter for repressor establishment, which is here. So PRM is going to be right next to C1. It's got this nice positive feedback loop. On the other hand, the promoter for establishment of a repressor is over here, further to the right, and in fact overlaps with the crow gene. Now, crow is being transcribed over here to the right. C1 transcribed over to the left. So if you have expression of this repressor establishment gene, what it does is it makes, the mRNA for C1, but also what it does is it makes an antisense RNA for crow. And what that means is you've got these two RNAs now, which would be binding to each other. Double-stranded RNA is not going to get translated. And so you also have a level of regulation here, even if there is any of that crow messenger RNA. If you're making this particular protein, gene, I should say, from repressor establishment, it's going to be blocking that by making double-stranded RNA. And how is that turned on? It's turned on by yet another one of these proteins that, when mutated, only gives you lysis, which makes perfect sense because you don't have C2. It's all going to be clear because you're not making your lambda repressor. But then again, how do you turn on C2? This is sort of like the Russian doll thing. Okay, it's all about C1. No, it's not about C1. It's about C2. It's not about C2. It's actually also about C3. And so that's our last thing down here. C3 regulates C2, and that's also part of this process here. You can the messy, this really sort of spaghetti diagram up here at the top. That's all that you need to know about this regulation, which again is rather messy. But it's about C3, and C3 somehow, and it's actually not entirely clear how C3 does this, basically responds to the metabolic state of the cell. And so if the cell is undergoing really rapid metabolism, that means the going is good. C3 is actually not present at all. If it's not undergoing a lot of metabolism, so there's not going to be a bunch of extra hosts to go to, then C3 is there. It stabilizes C2. You have C2. You start to express C1. Once you've expressed C1, you're good to go because you've got this nice positive feedback loop until you undergo induction. C2, as well, as a transcriptional regulator, blocks excisionase, which is important for integration that we'll get to in just a second. It also blocks expression of Q, just the same way as you're getting blocking of expression of CRO. It's this antisense RNA, and very conveniently called the promoter for anti-Q, PAQ, which will block expression of your Q protein. And of Q proteins, of course, is exactly what you need to get that final round of lytic replication, because that's that last anti-terminator that leads to expression of all of your late genes. Okay, so last part about all of these regulatory processes. This is why I said lysis is way easier to understand than lysogeny. So the C2 gene, which again is a transcriptional activator, activates at repressor establishment, PRE, and then antisense for CRO, antisense for Q, but then it also stimulates expression of the integrase gene. And the integrase gene, which we'll talk about in just a second, does what it sounds like. It's integration. And so it will take the viral genome and put it into the host genome. It's a site-specific recombinase, binds to a couple of attachment sites, both the attachment site in the bacteriophage and the attachment site in the bacteria. So these are the at sites, at P and at B, and we'll take a look at that in just a second. I also mentioned this before, but I wanted to repeat this. This cell, once it's a lysogen, stays a lysogen and is resistant to future infection because that lambda C1 is a repressor of everything else, and it's got too much. It's got more than's actually bound to the DNA. So any other, what they call superinfection, so more lambda coming in, will be blocked immediately because the lambda repressor is going to bind to PR and PL. So it's never going to be expressed. So it's just a competitive inhibition, but that particular, it never has a chance to make the genetic switch because there's already lambda C1, which is present inside the cell. Yes, happy, no? Ready for another clicker question? Of course, yes, we always are. So, let's see, now we need to turn this thing back on. Spending too long not asking you. I should ask you more clicker questions, right? Should make it easier. Decision by lambda to lyse or undergo lysogeny, actually say lysogeny, is mostly due to regulation of transcriptional initiation, transcriptional elongation, transcriptional termination, transcriptional initiation, transcriptional elongation. Now, of course, yeah, these should look really familiar because they're exactly the same answers we had last time. But is this answer the same? That is the question.
Look at this. They make it totally awkward.
You need to get my camera up there.
Okay, time to vote.
Slaps on. That's right.
The same way.
Yeah, it's about transcriptional initiation and transcriptional initiation where? If you're doing lysis, it's going to be initiating where? PL and PR. That's where you're getting your transcriptional initiation. If it's going to be undergoing lysogeny, it's going to be PRE for establishment of repressor. So yes, it is A. Okay, so let's talk briefly about integration. And this I've basically talked to you about already. It's the expression of the integrase gene right here, which is turned on by what? C2. So the C2 gene is causing expression of the integrase gene. This integrase gene, again, binds to the attachment site on the phage genome, that's at P, and the attachment site in the bacterial genome, which is at B. The attachment site B in the bacterial genome, as I mentioned before, this is a site-specific recombination. So it's always in this place, and it's between some of the galactose metabolic genes and the biotin metabolic genes. And so if you look at the genome of E. coli K12, you will find that between the galactose genes and the biotin genes is the whole lambda genome. Now, this lambda genome is now integrated in a linear form because you know the two circles. So you have this circle, which interacts with, in fact, this really big circle because, of course, it's a smart organism, E. coli, and has a circular genome. But it's just this little linear piece. It's a tiny little piece right here. So this integration event literally will take sequences here at the, also known as the operator site, but right in the middle here, and basically crosses these over. So this X here represents how this integration takes place. So this sequence P in the phage ends up being right next to the B prime sequence over at this end. And so you cross over here, link up this part of the Lambda genome with, now that's not important what the name of the genes are here, but J going back across, and then all the way around the genome on the other side, crosses across here, and you end up next to the gal gene. So now, this is the blow up, and I should have had that same image we had at the beginning. Once you have integration that takes place, that's that little piece, little brownish piece in our black genome. So these are how this happens. There's a what's called the ARM sites, and this is the integration site. As I mentioned before, the at-P site is much bigger. It's 250 nucleotides rather than 25 nucleotides, which is what you have down here. And that's because the integrase gene binds to this really big segment of DNA right here. It also has to have a cellular protein to bend this structure, and that's called the integration host factor, also known because of looking at lambda integration. Turns out IHF is also really important for transcriptional regulation of normal genes inside the bacterial genome, but was originally found in this case. And all of this stuff together, the integrase protein, the DNA, integration host factor, makes what all of us molecular biologists love to call things zomes. So the zome is nucleotides plus proteins, and it's that whole process which allows integration to actually take place. So integration, great. This is what happens, again, when you've got unhappy cells, high C3, high C2 expression of C1, but also expression of the integrase gene. On the other hand, sometimes you need to get back out, right? So if you have DNA damage, you turn on the SOS response. C1's not around anymore. Well, we're not going to talk about the regulation of those particular genes. If you want to read about them, they're in the textbook. They're in 4 of the chapters in this book, but we're not going to talk about it, so it's not going to be on the exam. But what happens is, under those induction conditions, you make some more proteins, and most important of those is this XIS. XIS just stands for excisionase. So excision, what does it sound like? Okay, it's going to be this excision process. And it's just the presence of this XIS protein which causes this process to take place. So excision of this circular genome from the cellular genome. In the absence of XIS, then you have integration that takes place. And so it's literally just the presence of this one protein that will lead to cutting it out or putting it back in. You still need the integrase protein because the integrase protein is literally just a recombinase. All it does is bind to and cut DNA and put it back together. And so that process is not going to be particularly directional one way or another because that's what the integrase does. It just binds to, cuts DNA, religates it, binds to DNA, cuts it, religates it. And so it's just the presence of excisionase which will lead to one direction or another. So we've talked about how you go from lysis, lysogeny, how you've got your integration taking place. This is all great, well and good, but we also need to make much more of our genome, right? So genome replication is pretty boring, not so ridiculously complicated. Good. So talk really briefly about the O proteins and the P proteins. These are expressed in the PR transcript, in fact, even before you get to Q. Origin binding protein, so pretty straightforward, O origin. And then the P protein, not so critical, but that recruits DNA B. What's DNA B? What was that course last term? We talked about replication like way before the first midterm in molecular biology. It's a helicase. Very most important thing that you need in terms of getting replication to take place is pulling those two strands apart. So pulling those two strands apart, That's what has to happen in order to get replication to take place. So early replication that happens in Lambda, you've got a nice circular genome. That circular genome has a normal origin of replication, separates, you have bidirectional replication, goes around the genome, standard 2 replications, forks go around the genome, get to the other end. You have termination that takes place. What happens, however, later on is you will switch from this theta replication to rolling circle replication. And we talked about this already in terms of Phi X174, and when I get the video running, we'll actually be able to see it. It's a lot easier to look at that way. But you're going round and round the genome, making many, many copies all together as concatemers. So linear copies of your genome attached end to end to end. And those then get processed and put into the virions. That process is here. We have our concatemers, so this represents multiple copies of genomes here, each of which is going to have this 12-base sequence that was found in the genome sequences, but Hershey showed was important then for getting these ends together. Many, many copies of this. There's a viral protein, not important what the names are here, called terminase. It's a terminase, okay, just terminates the end of one of these genomes. So terminase binds to the COS sites and literally will cut those COS sites in sort of the opposite way to when you have genome circularization that happens first thing on infection. So it will cut that and give you a genome with a terminase attached to a COS site going through to one more COS site. That's what you need to package your DNA inside this head structure. How do you get the head structure? The head structure actually forms in a way quite analogous to what we talked about already for Phi X174. Scaffolding proteins that form proheads or procapsid structures. So it's a structure which is not the final structure, and the way to get it is just assembly around scaffolding proteins. You need to lose those scaffolding proteins and then put the DNA into that structure. The DNA goes through a portal protein. Portal proteins should sound really familiar from, what was it, questions 49 and 50 on version A of the midterm. So this is the protein that sits at one of the vertices of your icosahedron, which is pumping the DNA inside the head. And once you've packaged the DNA inside the head, then you have a tail which gets attached to this. The formation of the tail structure is really fascinating. In fact, one of the authors of this book, Roger Hendricks, who's at the University of Pittsburgh, has spent most of his career studying this. Roger Hendricks got his PhD by a guy by the name of Watson. Sound familiar to any of you? So what he has been studying is looking at how this tail forms. And this tail formation is really fascinating. Turns out that there's one really long protein in the genome of the tail assembly structure. It's called the H protein. Again, the name is not important here. But it's one of the longest proteins, certainly the longest protein in the Lambda genome. And it turns out it's literally the length of that protein that measures out the length of the tail. So basically the tail assembles around this protein. Again, sort of a classic scaffold arrangement. And then once it gets to the end of the protein, it's done. And then the rest of it gets put together. The whole thing gets assembled. And once you have this assembly process, that you may or may not have remembered that there are lysis proteins that are being expressed by also the PR transcript. These are proteins that basically break down the peptidoglycan and make a hole in the external membrane of the cell. So, almost ready for our video, but before we do that, what do we have to do? Of course. So our last quick question for the day, I promise, is genome replication by Lambda is most similar to genome replication by Q beta 5174, bacteriophages C7, HIV1, or E. coli?
That's my first one. That's my gut.
I should say late genome replication.
Late. Okay.
We knew it.
Don't use that there. Please. Can I skip?
Yeah.
So if they're really professional,
Yeah, I just-
sort of
Hmm?
there are 80 protein, right, that sort of helps.
Mhm.
Is there a similar protein for that? Helping that?
The early replications, bidirectional, classic.
In terms of the replication process, we'll take a look at that when we get the video up and running.
Are we in agreement with
Yeah.
Yes.
Yeah. And it's
That's what I.
yeah. It's slide 15. So, early data. Which is kind of cool.
10. I think we got most of the votes already. It's good. Last chance to change your mind.
Yeah.
5.74 Y. Rolling circle, exactly. So I'm now going to try and play this DVD, which is now gray, and I really don't know why that is. So I'll see if it'll start here now, and if it won't, I'm going to switch it out and try and get it to play on this other one. It looks like that's not working. So I will also have this DVD on reserve in the library.